Welcome back, fellow mitochondriacs, for another episode of Cancer as a Mitochondrial Metabolic Disease. So, we have at this point covered a ton of information um, in terms of treating cancer as a metabolic disease. And the last, month at least, we've been talking about ways to lower glucose availability to cancer cells via whether it be a ketogenic diet, whether it be exercise, whether it be red light therapy, or a variety of foods and supplements to lower the glucose in the bloodstream. But now we're going to transition into talking about lowering blood glucose availability at the level of the cell itself. And that's going to require us to take a bit of a journey through the process of glycolysis, aerobic glycolysis, Warburg effect, etc. So today will be a little bit of an overview of how we're going to approach the next portion of the series. So let me pull up what I plan on sharing with you all today. So I started out with this graphic again to just give us a place to kind of reorient ourselves, because there's a lot of moving parts, and obviously we get pretty deep into the weeds as a part of this channel and a part of this series. And I just want to get us back kind of on track. So just to really step back even further than this graphic here, or where I'm circled here, we have talked about the ketogenic diet. We talked a little bit about calorie restriction. We've talked about ketone supplementation, and we talked a little bit about stress management. But kind of like now going a little bit further into this portion of it, although I think this is a great start that Seafried proposed in 2017, I think there's a ton more we could be doing, which is why we spent a lot of time and energy on things we could be doing to lower our blood glucose even further besides these two things, right? So that's when we talked about like red light therapy, exercise, supplementation, fibers, et cetera, on things that can lower our blood glucose. Because ultimately, what are these two things doing? These things are lowering blood glucose and lowering insulin levels. So you may ask, well, how does stress management does it? Well, we talked about it a little bit, how cortisol has an effect, catecholamines have an effect at raising our blood glucose without even eating. So that's I'm pretty sure that's why he puts this here at stress management. Whereas the ketogenic diet does that as well, because we're taking in very few carbohydrates that would raise blood glucose, or even to some degree protein. But where we have not got so far at all, well, we've talked about glutamine inhibition, obviously ad nauseum. But where we have not talked about at all is the inhibition of glucose. And that's where I plan on taking us over the next several weeks, until how long it takes to get through it. So going back to the basics of kind of non-tumoral cell physiology, metabolism, we have kind of a crossroads of where energy flows through. So when we have normal cells, we can be what they call metabolically flexible. We can use glycolysis to break down glucose and carbohydrates. We can use beta oxidation to break down fatty acids. We can break down a variety of amino acids that are known as glucogenic or ketogenic amino acids. And they're all going to get into the mitochondria in some way, shape, or form. And they're going to go through the TCA cycle or the Krebs cycle. And then those substrates that come off of the TCA cycle and or the Krebs cycle are going to get flown into the electron transport chain. And then the electron transport chain, which is requiring the intermitochondrial membrane to be intact and the proteins there to be intact, the so-called oxidative phosphorylation, or the electron transport chain, makes the majority of our energy with the use of oxygen, called aerobic metabolism. So just diving into a portion of this that we have not talked about much, and that is glycolysis. Okay. Glycolysis is how we take glucose and turn it into the next usable substrate by mitochondria, pyruvate. And there is a series of steps, as you can see here. And where there's a blue lettering here, this is an enzyme or a protein that converts the substrate that is in black to the next substrate, which is also in black. Okay, so if you could follow this along here, glucose is converted to glucose 6 phosphate via an enzyme called hexokinase. And then glucose 6 phosphate is converted to fructose 6 phosphate via phosphoglucose isomerase, and so on and so on and so on. And although my goal, as always, is not to make you a biochemist, I want you to be oriented on where we're trying to accomplish and achieve certain goals. And just to reiterate again, in terms of where we're at here, glucose, and this is a normal cell, glucose, let's say 1x, is being utilized and uptaked by these normal cells, and they're being converted through glycolysis to make pyruvate. Pyruvate is going to get into the mitochondria, because that's how it's supposed to work. It's going to get into the TCA cycle and then ultimately oxidative phosphorylation, and it's going to make ATP and some degree of ROS. However, in a cancer cell, we're going to use 10 to 30x the amount of glucose that a normal cell would uptake and utilize, signified by these very kind of fat green lines, right? And that is going to be utilized through glycolysis to make pyruvate. But because of a couple of things, we have things that are turned on within a cancer biology, things like hypoxia-inducible factors and pyruvate dehydrogenase kinase, which effectively block pyruvate from getting into the TCA cycle, because PDK will block pyruvate dehydrogenase complex. What happens is that by kind of sheer necessity, this road is blocked. So therefore, the only road that can be utilized next would be the fermentation of glucose, or the fermentation of pyruvate, to lactate. Okay, and there's an enzyme in here that's not shown called LDH, or lactate dehydrogenase. And so what we're going to do is we're going to try to immobilize this aberrant metabolism, also known as aerobic glycolysis, or the Warburg effect. We're going to try to stop this process from happening to the degree that it needs to, so that we can shut off its metabolism. Just looking at this from another perspective here, we have mitochondrial defects that are catastrophic to the point where we cannot make energy through the electron transport chain. There's very little or no energy being made by OXPHOS. So what happens is glycolysis is the main way that these cancer cells get energy, through the Warburg effect. So we're bringing in more glucose. Glucose is being utilized to make pyruvate. Pyruvate is being converted to lactate, and round we go. Lactic acid, or lactate, is a big problem. I have kind of a whole little micro series on lactic acid and the tumor microenvironment. But lactate is the end product of glycolysis, aerobic glycolysis, or the fermentation effect. Lactate is made here. Lactate is also, you see here, same process, just different graphic of way in showing it. Well, lactate's a big problem because lactate is what's at least partially responsible for the acidification of the tumor microenvironment, which ends up leading to immune suppression, immunotherapy suppression, metastatic spread, and chemotherapy resistance, and likely other resistance as well. So lactate is a big problem. In addition to that, what's not shown on that graphic is kind of how things come together. So we have the Warburg effect, which we've talked about so far, the pathologic glycolysis that's making lactate, that's acidifying the tumor microenvironment, which leads to chemotherapy resistance and immune resistance as well. But what is not shown on these prior graphics is that this Warburg effect is being programmed, at least by a large degree, via these hypoxia-inducible factors, HIF1 and 2. And that is driven by either hypoxia or pseudohypoxia. And again, we have videos on this, but hypoxia-inducible factors is at the crossroads of hypoxia, inflammation, and cancer. And it leads to tumor progression through altering metabolism, glucose and glutamine metabolism, creating new blood vessels, cancer stem cells, migration, invasion, tumor acidosis, and tumor-related inflammation, which again leads to tumor progression. And that can be driven by these HIFs can be driven by hypoxia or even pseudohypoxia, which again, we have a video series on. But to reiterate the importance of the Warburg effect and the metabolic reprogramming that happens under the control of the induction of hypoxia-inducible factor, the stabilization of hypoxia-inducible factor, we see that wherever there's a black lightning bolt or a red lightning bolt, we see here that it's upregulating glucose transporters, upregulating those glycolysis enzymes. It's upregulating lactate dehydrogenase, as well as we've talked about this in the past, it's upregulating glutamine as well. Glutamine uptake, glutamine utilization at multiple steps. So hypoxia-inducible factor is a major pathway that potentially could be a therapeutic of importance to us from a metabolic perspective. The other things that we've talked about in the past, things like KRAS, c-Myc, mTOR, AKT, PIK3, AKT, mTOR pathways, etc. Those are also going to be covered in this series because these hypoxia-inducible factors, MYC, PI3K, AKT, mTOR, AMPK, these all have influences on the upregulation of these pathways that are responsible for metabolizing glucose and converting that glucose into lactate ultimately. And so we're going to talk about this as part of this series as well. We're also going to talk about the role of pyruvate dehydrogenase kinase, which is under the influence of hypoxia-inducible factors, as well as MYC, and how we can influence that. We've talked about that. We've talked about this to some degree when we talked about dichloroacetate, but we may have to circle back and talk about that again. So the way I kind of envisioned looking at this problem is to be very systematic and methodical about it. So what I'll probably likely have is I'll have an entire video on the blockade or downregulation of these glucose transporters, look at its physiologic importance within cancer, and then look at what blocks them or downregulates them. In particular, I'll probably talk about both drugs and I'll talk about natural products that could potentially do this. And then I'll move on to the next video to hexokinase, phosphorfructokinase, pyruvate kinase, lactate dehydrogenase, pyruvate dehydrogenase kinase, glucose 6-phosphate dehydrogenase, which gets us back into the pentose phosphate pathway, which is an offshoot of glycolysis. We'll talk about hypoxia-inducible factors and how to potentially inhibit those, how to activate AMP kinase and activate sirtuin-1, which also have a key role, as well as the inhibition of PIK3, AKT, mTOR pathway, and the MYC pathway. And, you know, basically, I've had to, and I'm still having to, look through a lot of papers to kind of get us oriented, because what we'll see is over and over again, this will kind of be at least one of our probably classic graphics we keep going back to when it comes to the glucose side of things, kind of like we had that classic graphic that I would always refer back to when looking at the glutamine pathways. We have, you know, glucose transporters, we have, you know, hexokinase or pyruvate kinase or lactate dehydrogenase and PDK, hypoxia-inducible factor 1 alpha. And what you'll see is you'll see a bunch of things, right, that are potentially having an effect on that, or another kind of grouping of things that have an effect on this, or have an effect on this, or vice versa, or so on and so on. And it gets a little bit dizzying at times. And so we'll look at each of these steps, and I'm going to try to look at multiple papers, not just this one paper, because although this paper has a great graphic, associated with it, and kind of gets us oriented. Paper number next may have a graphic that looks very similar, but is got different compounds, right? And then there may be another graphic that looks very similar, but has different compounds looking at different pathways. And then there may be another paper that has, again, a very similar, kind of orientation. We have the glycolysis pathway and the same kind of key enzymes that are being inhibited, but may have a bunch of different other things. So my goal with this is to get organized, because these tend to be pretty all over the place in terms of standardization, I guess you'd say. So we're going to try to bring some order to the chaos. And then we're going to look at, for example, the pentose phosphate pathway, which is also a key pathway that is upregulated and utilized in cancer metabolism and Warburg metabolism. And then we're going to look at AMPK and how, when AMPK is downregulated or inactivated, you may say, it has effects on stabilizing hypoxia factor, which leads to the Warburg effect, or, as I had also mentioned, sirtuin-1 having a crossroads where it's also, when it's downregulated, will stabilize hypoxia factor, which will lead to the Warburg metabolism. And what we'll end up doing is we'll try to piecemeal this together. Now, you may be asking, if you're just now coming in to this part of the series, why is this important? Well, we've kind of covered it here, right? We kind of talked about how these cancer cells require 10 to 30x more glucose than a normal cell. We've also kind of touched on it without saying it. This is where the PPP comes off right here: biosynthesis and proliferation of cancer cells. It's just very vague and it's not very detailed, this graphic. And we have talked also at length about how these two molecules are in particular of particular importance. This is a Seafried graphic that I stole from one of his most recent papers. I want to say it was like end of November, beginning of December 2024. So kind of hot off the presses here. And we see here that these two molecules are of critical importance for the proliferation and continuation of cancer, uncontrolled cell growth, and ultimately disease progression. And what we've covered this kind of at nauseum, things that inhibit either glutamine uptake or glutamine utilization. And here we've touched on, how glutamine is responsible for, at least partially responsible for, making new DNA and RNA, and fatty acids through citrate, and other proteins and other amino acids. We've kind of touched on how glutamine is responsible for being fermented to succinic acid, which is the other kind of big part of the extracellular acidification process. And we've definitely touched on, glutamine's role in the creation of glutathione. That we have talked about a lot when we talked about ferroptosis. But what we have not talked about as much on this series is how glucose is the other end of the spectrum or the other very important part of how these cancer cells become out of control growth. We know that glucose is fermented to make lactate to make ATP. That's something that I think is well talked about. If you listen to Dr. Seafried's podcasts or lectures that he gives, we know that it makes ATP. But again, and I'm not saying he doesn't know this, he clearly knows this. This is his graphic from his paper, but he doesn't talk about it much, is how glucose contributes to fatty acid synthesis or amino acid synthesis or glutathione synthesis or nucleotide synthesis. And we want to shut this process down to the greatest degree possible. From another way of looking at it, we have glucose being uptaked into these cells, right? Another Seafried's graphic, taken up to these cells, and they can either go through the Warburg effect or aerobic glycolysis to make pyruvate, and then ultimately lactate to make energy. But glucose can also be siphoned off at the level of glucose 6-phosphate to go into the PPP or the pentose phosphate pathway to make nucleotides, to make glutathione, et cetera, et cetera. And so what we're going to be talking about is ways to shut this process down at multiple levels. So going back to this graphic here, we're going to be looking at glucose transporters first, then hexokinase, then pyruvate kinase. I think there's also going to be another lecture on phosphofructic kinase. There's going to be lactate dehydrogenase, pyruvate dehydrogenase kinase, hypoxia factors. We'll talk about glucose 6-phosphate dehydrogenase, or G6PD, ways that we can potentially have influence over that. We'll talk about a lot. We're going to talk about a lot, as you can imagine. So these are going to be the way that we're going to attack the problem. We're going to break it down into specific enzymes. We're going to talk about those enzymes' particular importance in the continuation or the progression of cancer. And we're going to talk about basically either drug things or natural things that can have influence over either down-regulating or blocking these pathways. So today will be a shorter video because I want to set us up for success the next several videos. And let me see if I can't go to some of these questions. How do they make these beautiful illustrations? I wish I knew, but I'm glad they have them. They're all pulled from papers from, in terms of if they're done by AI or people who specialize in graphic design. My gut tells me it's probably people who specialize in graphic design for biology papers, because some of these papers are pulled from, the early 2000s or mid 2000s. And so I think that, I don't believe we had AI at that time. So I think it was probably just people who were specializing in graphic design. And what's cool is that over time, those graphics just keep getting better and better. One question I have is, does cancer require insulin to utilize glucose? No, I do not believe insulin is required. If insulin was required, then we would just stop cancer completely with a ketogenic diet, which does not happen generally. I'm not saying it's not an important piece of it. A lot of the upregulation of glucose and glutamine transporters and machinery do not require insulin. They're under the influence of hypoxia factors, MYC, RAS, etc. And then some of the other pathways like mTOR and PIK3 AKT. That's one way insulin will work through those pathways, but it doesn't require insulin. Definitely insulin is throwing gasoline on the fire for sure, because insulin will upregulate those things as well and create a very anabolic situation. And then someone had mentioned, yes, glucose restriction with diet. I have cancer now 4 years and use glucose restriction through diet. Carnivore cancer is stalled out B. Not sure what that lastly means, but yeah, so definitely just, quickly going back to reorient ourselves. Just quickly reorienting ourselves back to the beginning of how we started this live stream today. So definitely a ketogenic diet and lowering availability of blood glucose is critical. There's no question. That is the foundation. Lowering stress and lowering our amount of glucocorticoids like cortisol and catecholamines that can raise our blood glucose, that also is critical to lowering availability, right, of cancer to have the 30 to 60x, or excuse me, 10 to 30x times more glucose. If we can lower the amount of glucose coming in because there's not available glucose, that would be one way to do it, right? But where we're talking about over the next several weeks, and particular maybe months, is talking about ways to inhibit glucose utilization and uptake so that we can actually ultimately shut this whole process down. When we shut down glucose uptake because there's no glucose available, because if we're on a low carbohydrate or ketogenic diet, that's one way. And then blocking several enzymes along these paths, that way we can't have the fermentation of lactate. Then what's cool is that the lactate tumor microenvironment will then collapse, and the immune system can then turn on, and our and our other therapies that maybe we may be utilizing with the help of conventional medicine, whether it be chemotherapy, immunotherapy, et cetera, can also have a better effect. And then because we are taking care of the glutathione issue, then things like HBOT or IV vitamin C or other oxidative therapies like photodynamic therapy, even radiation therapy, and even chemotherapy, depending on the type of chemotherapy it is, will work by creating oxidative stress and ultimately leading to cell death. So again, this is the general recipe, right, for success. We have ketogenic diet, we have stress management, we have glucose and glutamine inhibition. And then the last piece of the puzzle, which we'll spend a significant amount of time on, will be oxidative therapies. And oxidative therapies, at least on this particular graphic, is going to be hyperbaric oxygen therapy. And I'll just kind of maybe end by saying that it's very unlikely that any one of these pieces of this puzzle will work in a vacuum. Just like, if you do a ketogenic diet alone, you'll have some success likely, but not complete winning of the war, right? If you manage your stress, you're going to have some success, but not winning of the war. If you start to block glucose and you're not in ketosis, that could be problematic, and you're probably not going to win the war. If you block glutamine by itself, if you just take Dawn or, I don't know, mebendazole or EGCG or something like that, and you're just blocking glutamine by itself in a vacuum, you're not going to have success. If you do HBOT or IV vitamin C or photodynamic therapy by itself, you're probably not going to have success. But when you start to use this as a system and you start to break down all of the Achilles heels that are associated with the Warburg effect and how these cancer cells metabolize all these substrates to help protect themselves, to power themselves, and to grow, that's when we start to be able to manage and hopefully, God willing, resolve the disease process over a period of time. And I hope that at least if you've been with me for a while, you can start to see the beauty in this very simple graphic. Now, taking this simple graphic that a brilliant researcher and his team put together and taking it and converting it into clinical medicine at the level of the patient at the bedside, that is another story. That is going to take time to kind of hash out all the details. But we have a beautiful framework here that we're going to be working off of. And we finally hit the portion of the series where we're talking about glucose inhibition, uptake, and utilization. So I hope that this makes sense of why this is important. Got a question here. Does the HBOT specific have to be a hard chamber, or do soft chambers have any value? Well, we'll talk about that when we talk about HBOT. But what I can tell you is that the ideal situation is that you're in a hard shell HBOT chamber. But does that mean, to answer your question, that the soft chambers have no value? That is not true. The soft chambers do have value, and I can tell you, and I'll probably have to have him on. He agreed that he would be on the channel as an interview. But Dr. Dom D'Agostino, I had emailed him, and he believes that a soft chamber can be used, just needs to be for a longer period of time than what would be normally recommended at a higher atmosphere or ATA. So hopefully that answers the question. Anyways, I hope you are, looking forward to this portion of the series. I think that this is where it gets pretty interesting, and we get to use a lot of tools in our tool bag for this next piece of as well. What I also would say is that a lot of the compounds and supplements and drugs will dovetail into other portions of the strategy. For example, let's say curcumin. Curcumin has profound effects at lowering blood glucose. Curcumin also has effects on lowering glucose uptake and utilization. It also has effects on glutamine uptake and utilization. So therefore, curcumin, even though it will have effects kind of in the vacuum that we're looking at it as whether it be like a hexokinase or a pyruvate dehydrogenase kinase or whatever inhibitor, it will also have diverse effects, which means that we can use one substance or a couple of handful of substances that can have effects on all of these pathways, which is pretty unbelievable. If you like videos like this, if you like where we're going with the channel, if this is helpful for you or someone that you love, please like the video. If you are not yet a subscriber to our mitochondriac army, please consider subscribing. We would appreciate it. And until next time.